Hi guys, my name is Jamil Ahmed and this is another tutorial about biochemistry. Welcome back. As you know that we were discussing about the transcription, and in this lecture we will discuss about the alternative splicing of eukaryotic pre-messenger RNA. As you know that in case of eukaryotic transcription, the messenger RNA formed is the pre-messenger RNA. It's not a mature messenger RNA. It undergoes some processing, and after this processing it will be changed into mature messenger RNA, and then mature messenger RNA will move outside from nucleus to cytoplasm, where translation will happen and proteins will form. And in this lecture we will discuss about the alternative splicing of eukaryotic pre-messenger RNA. That what is the alternative splicing? First we must know that what is splicing. You know about splicing, but I would like to repeat about splicing. So you know that in case of eukaryotic transcription, the messenger RNA formed is the pre-messenger RNA. It's not a mature messenger RNA, because in pre-messenger RNA, in eukaryotic transcription, the messenger RNA formed is the pre-messenger RNA because it contains introns and exons both. Exons, which are the expressed parts, are the coding regions which have information for the synthesis of proteins, and introns are the intervening regions, or we can say that non-coding regions which doesn't have any kind of information for the formation of proteins. So exons are interrupted by introns, and first we have to remove these introns by some process. That process is called splicing. So splicing is the post-transcriptional process. Post-transcription means that it happens after transcription is complete. Post mean that after. So after post-transcriptional process means that when transcription will be completed, then after that transcription, after that splicing will happen. Means that it's a post-transcriptional process in which introns are removed. As I said earlier, that in case of eukaryotic transcription, the messenger RNA formed is the pre-messenger RNA because it contains introns that are between the exons, between the coding regions. We have to remove these intervening regions, we have to remove these non-coding regions which are between the coding regions. So these introns are removed, and exons, which are the expressed parts, or the coding regions, are joined together. Okay, so these exons will join together. Let's suppose in this way, let's suppose this is a one exon, this is a second exon, until here there are exon. So this is one exon, this is a second exon, and between the exon, if we suppose that between the exon, this one is the intron, then these are the non-coding regions and these are the coding regions. We will remove this exon, we will remove this intron. Okay, this intron will be removed, and then these exons will join together. So these exons will join together. So this exon will join together in this way. So this this exon will join together. So now in this way, this is called the splicing, and in the result of splicing, the pre-messenger RNA will be changed into mature messenger RNA. Okay, I think now it's understandable for you the definition of splicing, that this is the post-transcriptional process in which introns will be removed and exons will join together to form the mature messenger RNA. This is the definition of the splicing. Now we will move on and we will see that what does mean alternative splicing. Here is the alternative splicing. What is the alternative splicing? In alternative splicing, primary transcript, or we can say that pre-messenger RNA, immature messenger RNA, is spliced differently. It is spliced different way to produce many different variants of proteins from same gene. What does it mean? I would like to say that I would like to explain it in a simple way, in a simple idea. We can write the simple idea. What is that simple idea? We can say that a simple idea. So if we write the simple idea, then we can say that same gene or a single gene, transcription will happen, and it will change into a single pre-messenger RNA. In case of eukaryotic transcription, so pre-messenger RNA will be formed, and this pre-messenger RNA, or single messenger RNA will be spliced in a different way, spliced different ways and will produce the different proteins. Will produce the different proteins. What does it mean? It says that same gene, one gene, then transcription will happen, it will form the pre-messenger RNA, that is the single same pre-messenger RNA. Then this messenger RNA undergo splicing process, in which these introns, let's suppose that this is a single pre-messenger RNA. Let's suppose this is a single pre-messenger RNA which is obtained, which is synthesis by the same gene after transcription. Okay, so let's suppose this is the same pre-messenger RNA which contains exons, introns. So this is a pre-messenger RNA because information is fragmented. Exons are interrupted by introns, exons are interrupted by introns, exons are interrupted by exons. So this is a single pre-messenger RNA. Then we have to then here will happen some process that is called the splicing, because this is a pre-messenger RNA. What does mean splicing? Mean that these introns will remove outside and these exons will join together and they will form the mature messenger RNA. So splicing will happen, mean that these introns will remove. But in alternative splicing, this splicing will happen in different ways. What does mean? It does mean that at some time this intron will remove and exon A, B and C will join together. Okay, C will join together and they will form the one kind of protein. And in another kind of splicing, this intron 2 and 4 will remove, this intron will remove and exons A, C and D will join together and they will form another protein. Remember, just focus on this concept. Let's suppose that this is the pre-messenger RNA. Then it will splice in a different way. Let's suppose that first this intron will be removed outside. So this intron will be removed outside. Okay, so when this intron will be removed outside, then A and B, let's suppose A, B, C, D exons, 1, 2 and 3 introns. Okay, so first intron will remove outside. Then after that what will happen? A and B, these exons will join together. Okay, A and B exons will join together, and let's suppose that at the same time this intron 2 is also spliced outside, removed outside, then C will also join with these. So A, B, C mean that exon A, exon B and exon C will join together, and they will form the one kind of protein, because they will form the amino acids and amino acids will form the one kind of protein. Okay, one kind of protein. Okay, now this concept is okay. Now we will move on. In the next thing, if you see in this kind of splicing, this is the one kind of splicing, and in another kind of splicing, another introns will remove and another 3 exons will join together. Let's suppose at this time A, C and D exons will join together. A, C and D. In the previous setup, A, B, C, and in this way A, C, D. So exons are joining in a different way. In the previous A, B, C, there was a different sequence of exons, and here is a different sequence of exons. A, C, D. First was the A, B, C, but this time are the A, C, D. So different sequence of exons because of different splicing. So different introns will be removed and different exons will join together, and there will be the different sequence of exons. When there will be the different sequence of exons, then there will be the different variants of proteins. So this will be the another kind of protein. This is another kind of protein. In the same way, now if there is a collection between the B, C and D, so you can see B, C, D, so it is different sequence of exon. This is another sequence of exon, so it will form another kind of protein. Okay, now you can see that this protein has different sequence of exon, this protein has different sequence of exon, this protein has different sequence of exons. So all 3 proteins has different sequence of exons. Okay, the different sequence of exon, different sequence of exon, different sequence of exon. So proteins are also different. So in this way, splicing will be different. When splicing will be different, then different sequence of exons will join together. When different sequence of exons will join together, then there will be different variants of proteins. I would like to give another concept here, that when there is a different sequence of proteins, then the structure of proteins will change. As you can see here, that if we are going to join the A, B, C, this is a different structure. A, C, D, different structure. B, C, D, different structure. When their sequence of exons will be different, then structure of the proteins will be different. When structure of the proteins will be different, then automatically their functions will be different. So we can say that from a single gene, let's suppose there is a single gene, from single gene, single pre-messenger RNA, from a single messenger RNA, we can form the variants of proteins. You can see that different protein, different many proteins, we can form the many proteins. Same this definition says that primary transcript, primary messenger RNA is spliced differently. It will splice in a different way. Different electrons will remove and different exons will join together, mean that there will be the different sequence of exons to produce many variants of proteins from same gene, from same messenger RNA, pre-messenger RNA, we can form the many variants of the proteins. Okay, now we will move on. Next is the examples. So now we will talk about the examples of alternative splicing. The first example is membrane immunoglobulins versus secreted immunoglobulins. First we must know that what is membrane immunoglobulins and what is secreted immunoglobulins. Membrane immunoglobulins, these are the antibodies, these are the immunoglobulins, or these are the antibodies. Okay, these are the antibodies, or we can say that these are the immunoglobulins which are produced by immature B cells. Okay, these are produced by immature B cells. So these are the antibodies which are produced by the immature B cells and present on the surface of immature B cells, and when any foreign body, any foreign antigens comes, then these antibodies are produced by immature B cells to regulate the immune response. So this was about the membrane immunoglobulins, mean that what does mean membrane immunoglobulins. So second is the secreted immunoglobulins. What is the secreted immunoglobulins? Secreted immunoglobulins, these are also the antibodies, these are also the antibodies, but these antibodies are produced by the mature plasma cells, produced by the mature plasma cells. As you know that B cells are the immature, then these immature B cells will be changed into mature plasma cell, then mature plasma cells will produce the antibodies when they will see the any foreign antigens, then they will form the antibodies, and these antibodies will cause the regulation of immune response. So what is the difference between membrane immunoglobulins? These are the also antibodies but produced by the immature B cells, and secreted immunoglobulins, these are also the antibodies but are produced by the mature plasma cells. So we can say that these both are the immunoglobulins, these both are the antibodies. So now what is the concept of alternative splicing in membrane immunoglobulins versus secreted immunoglobulins? So these both immunoglobulins, either membrane immunoglobulins or secreted immunoglobulins, both are produced by a single gene, both are produced by a same gene. We can say that these both are produced by the same gene, these both are produced by the same gene. Okay, same gene, then same gene will be changed into same pre-messenger RNA. Okay, there will be the same gene, then this same single gene will be changed into the same pre-messenger RNA. Okay, now this pre-messenger RNA will undergo splicing in a different ways. Splicing will undergo in a different ways. What does it mean? It does mean that different exons will join together. And different introns will be removed. So on the base of this, same gene, same pre-messenger RNA, and there will be different splicing in a different ways, and different exons will join together. And if exons join in such a manner, in such a sequence, that the forming antibodies are hydrophobic, then it will form the membrane immunoglobulins. And in the second way, that splicing happens in this way, that exon sequence is in this way, that splicing happens in this way and exon sequence is in this way, that the forming antibodies are the hydrophilic, then in this way the forming antibodies will be the secreted immunoglobulins. You can see that both membrane immunoglobulins and secreted immunoglobulins both are produced by same gene, same pre-messenger RNA, but they are different from each other. So these are the different variants of proteins. From same gene we are producing the different proteins. If exons join together in such a sequence that they will form the hydrophobic, then these immunoglobulins will be the membrane immunoglobulins that are produced by the immature beta cells, and exons sequence joined with in such a way that antibodies are the hydrophilic, then these antibodies will be the secreted immunoglobulins, and these are produced by the mature plasma cells. Okay, so next example is the dopamine receptors. First you must know that what is the dopamine receptors. Dopamine receptors, these are the receptors in brain. These are the receptors in brain, or you can say that in central nervous system. Okay, so now dopamine receptors are G protein coupled receptors. These dopamine receptors not of one kind, these are of many types, many kinds. Okay, these are of many kinds. So what is the function of dopamine receptors? Different hormones come, let's suppose that A hormone, let's suppose that B hormone, different types of hormones comes, and C, D hormones, many hormones. These hormones comes, and these hormones acts on the dopamine receptors. Okay, different, because there is a many dopamine receptors, each dopamine receptor is specific for specific hormone. Okay, it is specific for specific hormones. So different hormones will come, and they will attach on its specific dopamine receptor. Then this dopamine receptor will activate the specific effector. Okay, so I would like to say the simple idea that there are many hormones that are acting on the dopamine receptors, there are many dopamine receptors that are receiving the many hormones, and there are many factors that are getting signal from many dopamine receptors. I would like to say that there is a many dopamine receptors which are different in structure and different in function. Okay, but they all are dopamine receptors. But they are different in structure, they are different in function. So also about now I would like to say about alternative splicing in dopamine receptor. In case of dopamine receptors, there is a same gene once again, same gene will form the same pre-messenger RNA, and this same pre-messenger RNA will undergo splicing, and there will be the different in the sequence of exons, then there will be the different in the structure of the dopamine receptors, and there will be the difference in the function of the dopamine receptors. These all are dopamine receptors are formed by the same gene, single pre-messenger RNA, but there is a different sequence of exons because of different splicing. Then there will be the different structure of proteins, different structure of dopamine receptors, and different function of dopamine receptors. That different hormones will act on the different dopamine receptors, and different dopamine receptors will activate the different effectors for taking action. Okay, so this was about the dopamine receptors. Next is the tropomyosin variants in muscles. Okay, first you must know that what does mean tropomyosin. You know that tropomyosin helps out in muscle contraction. No, here is a question, what is the muscle contraction? You also must know a little bit about physiology, that muscle contraction happens that when there is a two proteins in muscles, actin and myosin. You know that actin plus myosin. There is a two proteins in muscles, actin and myosin. When these actin and myosins join together, there is a cross bridging between actin and myosin, then muscle contraction happens. But what is the function of tropomyosin in muscle contraction? So now this tropomyosin comes, and when there arrives a signal that now we have to prevent the muscle contraction, then this tropomyosin will go and it will cover up the actin. When it will cover up the actin, then there will be the no cross bridging between actin and myosin. When there is a no cross bridging, then there will be the no muscle contraction. Okay, so muscle contraction, actin and myosin will cross bridge and muscle contraction will happen. But when we get a signal that now we have to prevent this muscle contraction, then tropomyosin will go and it will cover up the actin. Then there will be the no cross bridging between actin and myosin. Then there will be the prevention of muscle contraction. Okay, but here is a question that what is the role of alternative splicing in tropomyosin variants in muscles? So now this tropomyosin have different variants in muscles, in same muscle it has different variants. Tropomyosin has different examples: tropomyosin 2, tropomyosin 6. So these are different variants of tropomyosin. This is just because of the splicing, alternative splicing. So what does it mean? It means that there will be the same gene. Okay, once again there will be the same gene, there will be the same pre-messenger RNA. Both will be the same gene, same pre-messenger RNA, but the same pre-messenger RNA will be spliced in a different way. Then there will be the production of different variants of tropomyosin in muscles. Examples are the tropomyosin 2, tropomyosin 6. So these both are the tropomyosin formed by the same gene, but they are different in structure and both have different function. Okay, so this was about the tropomyosin. And next thing is importance. What is the importance of alternative splicing? What is the importance of alternative splicing? As we say that in this way we will form the many different proteins from single gene. So it does mean an organism can make many more proteins than genes to encode these proteins. Mean that if we have one gene, then from one gene we can form different proteins. Then the formation of proteins in an organism will be more as compared to genes. We need less number of genes, but there will be the more formation of proteins. Understand? Let's suppose that. Let's suppose this is so simple that if one protein, one gene, it will produce 3 proteins. Okay, 1, 2, 3. Another gene, it will produce 4 proteins. Okay, 1, 2, 3, 4. Third, it will produce 2 proteins. So now you can see that 1, 2, 3 genes, but producing 4, 3, 7 to 9 proteins. Therefore, here is the thing that an organism can make many more proteins than genes. Sorry, not that. Here will become the then genes to encode. You can see that genes that are encoding the proteins are just 3, but the forming proteins are the 9. So 3 genes and 9 proteins. Therefore this line says that an organism can make many more proteins than genes to encode it. And in the current situation in humans, you know that there is a 1 lakh proteins. There is a 1 lakh proteins in humans, but only 20,000 25,000 genes in our body. So this is just because this phenomena is just possible because of the alternative splicing. These 20,000 25,000 genes will undergo alternative splicing and they will form the different variants of proteins. Therefore proteins of number will be greater. The next thing, it's all about the difference from polycistronic. What is the difference between polycistronic? What is the difference between the polycistronic and the alternative splicing? Okay. Here are some concepts about polycistronic and alternative splicing. The first one is the many genes are transcribed and translated to different proteins. Remember the concept about polycistronic, that is the criteria of prokaryotic transcription. That in prokaryotic transcription what happens, that there will be the many genes, in this way it's a single gene, but in case of prokaryotic transcription there may be the many genes will be transcribed together. Many genes will be transcribed together and then they will be translated together into different proteins, because there is a many genes, many proteins, but they are transcribed and translated at the same time. Okay, but in this way there is just a single same gene. Here is a many genes, many proteins, but here is a single gene to different proteins. Okay, so in this way many genes were transcribed and translated at same time, but in this way a single gene is spliced differently. mean that in this way a sequence of exons will be different to different proteins. Here is a different proteins, here is a different proteins, but here are many genes and here is just a single gene. Many genes undergo simultaneously transcription and translation, but in this way a single gene undergo splicing, mean that different sequence of exons. And next thing, that many genes, because you know that there is a many genes, so many start and stop codons in transcription. You know that each gene has its own start and stop codon in transcription. Remember the concept in transcription I explained in the previous lectures, that in transcription each gene has its own start codon that starts the transcription and stop codon that stops the transcription. So because there is a many genes, then there are the many start and stop codons in transcription, and in this case, because you know that there is only single gene, so single stop codon and single start codon, so simple. And the next one, you know that this is a prokaryotic property, polycistronic. Eukaryotic transcription is only on monocistronic, but the polycistronic is the prokaryotic property. Polycistronic is only the prokaryotic property, but monocistronic is the eukaryotic property. Mean that prokaryotic transcription may be monocistronic, may be polycistronic, but eukaryotic transcription is only on monocistronic. And this polycistronic is the prokaryotic property, and you can see that alternative splicing happens in the eukaryotic transcription, because you know that pre-messenger RNA will form only in eukaryotes, not in prokaryotes, and pre-messenger RNA undergo alternative splicing. Therefore, alternative splicing is the property of eukaryotes. And one more concept, one more important concept about the alternative splicing, one more important concept. Let me to clean this one, splicing, and let me to explain it right out here. Here is a one concept. That concept says that, what is that concept? That concept says that this alternative splicing may happen in a single cell. Okay, in a single cell, or we can say that in the same tissues, or alternative splicing may happen in the different tissues as well. So what is this concept? I am going to give a little bit about this concept. So alternative splicing may happen in the single cell, or we can say in the single tissue. As you can see that antibodies, same tissues, same cells, so it are going to form in the same tissue, membrane immunoglobulins, secreted immunoglobulins, both are formed in the white blood cells, same cells, both are the same cells, same antibodies, same beta cells, either mature beta cells or plasma cells, both are the same cells. Okay, so these are formed by the same cells in a single cell, single tissue. These are examples of that one. But this alternative splicing may happen in the different tissues. This alternative splicing may happen in the different tissues. What is the example of that one? You know that if you talk about the tropomyosin, once again, example of tropomyosin. Once again, example of tropomyosin. So you know that tropomyosin in different tissues. Smooth muscles, we are going to talk about two things: smooth muscles and skeletal muscles. This tropomyosin has different structure in smooth muscle and in skeletal muscles. These tropomyosin is formed by the same gene, same pre-messenger RNA, but when it goes in the smooth muscles, then there is a different sequence of exons, and sequence of exons is in this way. Then the tropomyosin formed. You know what is the function of tropomyosin? It covers the actin. Then when tropomyosin when the gene of tropomyosin goes in the smooth muscle, then exon sequence is in this way. Then tropomyosin variant, different variant, it will cover up the 14 actins. But when this gene of tropomyosin goes in and the tissue, that is a skeletal muscle, it goes there, then there is a different sequence of exons, and this variant of tropomyosin will cover the 5 to 7 actins of 5 to 7 actins. Okay, now you can see that tropomyosin in different muscles, smooth muscles and skeletal muscles. When it is in smooth muscle, it covers 40 actins. When it is in skeletal muscles, it covers 5 to 7 actins. Mean that it has its own different splicing. When there is a different splicing, when there is a different sequence of exons, when there is a different sequence of exons, then there is a different structure. When there is a different structure, then you can say that there is a different function. So I am trying to say that alternative splicing may happen in the single cell, as you can see that in the this is a single cell, same cell, and may happen in the different tissues, as you can say that smooth muscles and skeletal muscles as well. In smooth muscle, it covers 14 actins, mean the different variant, and in skeletal muscles, it covers 5 to 7 actins. And it has its own, meaning that it's a different structure and different function. So it was all about the alternative splicing of eukaryotic pre-messenger RNA. If you have any question about this topic, alternative splicing of eukaryotic pre-messenger RNA, then you can comment me, you can ask me questions and I will answer you. So thank you so much guys, see you in the next lecture.